everyone and welcome to Miss Estruch Biology, and in this video I'm covering all of topic 2 for AQA A-level Biology. And if you didn't know, you can download for free by clicking the link in the description the workbook that I created that goes alongside this entire video. So save yourself time having to make extensive notes and do this instead to get a general overview. But if you want even more detail and a better idea about key terms, examiners tips and topic summaries, then check out my ready-made A-level notes or my flashcards. But that is it for now. Enjoy the video. AQA topic 2, the entire topic. Let's get into all of this information then, starting with 3.2.1, which is cell structure. For the eukaryotic cells, these are the 10 organelles that you need to know both the structure and how that structure links to the function. So we're going to go through all of these. Here's just showing you an overview though of what they look like inside of the cell. So we have our cell membrane on the outside. Now, although it does say vacuole here and it's an animal cell, animal cells don't have permanent vacuoles, but they can have temporary vacuoles, things like your phagosome that you get in phagocytes, but we can kind of ignore that for the sake of this slide. Rough endoplasmic reticulum, so you've got these folded membranes with the ribosomes on the outside, so they look rough or bumpy. Golgi apparatus, the one that looks like a Wi-Fi symbol. The cytoplasm, lysosomes, ribosomes, which are the smallest, and then smooth endoplasmic reticulum and the mitochondria. So centrosome is not on the AQA spec. In plant cells, they have all of those structures plus 3 additional ones. They do have a permanent vacuole, which is surrounded by a membrane called a tonoplast. They have the chloroplasts, which is the site of photosynthesis, and then they also have the cellulose cell walls. So if we have a look at the nucleus first of all, the structures and the functions. The nucleus has got an envelope, which is this membrane around the outside, this double membrane, and there are holes within that called nuclear pores. The liquid in the middle, or the jelly-like material, is the nucleoplasm. You have the chromosomes within the nucleus, which are protein bound and described as being linear DNA. And then we've got the nucleolus, which is this additional small sphere inside of it, which in terms of the function, really that should be on that side, but the function is the site of RNA production, and it makes the ribosomes. So if you were asked to describe the functions of the nucleus, one of them we've already said down here, RNA production, and it makes ribosomes. It also contains all the genetic code for each of the cells, and it's the site of DNA replication and transcription, which is the making of mRNA. Endoplasmic reticulum can be rough or smooth, and both of them are made up of folded membranes called the cystinae. But the rough has ribosomes attached to the outside, which is what makes them rough. So here it is under the microscope, and all those tiny black dots are the ribosomes, giving that rough feel or rough look. The function of the rough endoplasmic reticulum is for protein synthesis, and the smooth endoplasmic reticulum is the synthesis of the lipids and carbohydrates. The Golgi apparatus and then the Golgi vesicles that pinch off. First of all, the structure though, it's folded membranes making these cystinae again, and it creates this Wi-Fi symbol is what I hear most people describe it as, because you've got these curved folded membranes and then the vesicles bubbling off. So those secretory vesicles pinch off from the cystinae. There's a whole range of functions for the Golgi apparatus and the vesicles. So you could have carbohydrates being added to proteins to form glycoproteins. You also get secretory enzymes being made, secrete carbohydrates. They transport, modify and store the lipids. They form lysosomes. Molecules are also labelled with their destination here, meaning you might add on some sort of chemical tag so it can only bind to a target cell. And also we've got the finished products are transported to the cell surface through these Golgi vesicles. They move up and fuse with the cell surface membrane, and that then releases the contents of the vesicle. The lysosomes are these bags of digestive enzymes. You can see one here. So just like sacs filled with digestive enzymes. So all of their functions are going to be to do with hydrolyzing and digesting. So they could be hydrolyzing pathogens inside of the phagosome. So in your phagocytes in topic 2, which we'll get into later on in this video, you've got your phagosome that has got a pathogen engulfed within it. The lysosome contains digestive enzymes, it fuses with the phagosome, and that digests the pathogens. It could be to completely break down dead cells within the body that you don't need anymore. You could have exocytosis, meaning the release of those digestive enzymes to outside of the cell to destroy material. And also it might be to digest worn out organelles so that you can reuse those materials. The mitochondria is a double membrane bound organelle. We've got the outer membrane here, and the inner membrane is folded. It's shown as this bright yellow structure here, folded so that you've got a really large surface area for some of the stages of respiration. So that folded inner membrane is called the cristae. Inside of that you have a fluid-filled centre, which is known as the matrix. You learn more about this in topic 5 when you learn aerobic respiration. You also have loops of DNA and 70S ribosomes, which are the smaller ribosomes that you typically get in prokaryotic cells. And you need both of these structures so that within the mitochondria, you can have the creation of proteins, for example, enzymes that are needed for aerobic respiration. So the function then is it's the site of aerobic respiration, not anaerobic, just aerobic, which means you have lots of ATP being produced here, and also we get enzymes being created for respiration. Ribosomes, these are small, they're the smallest of the organelles, made up of 2 subunits of protein and RNA. Those are the 2 molecules that a ribosome is made up of. You have 2 different sizes, ATS, which are the larger ones, which are found in eukaryotic cells, and 70S are smaller in size, which are found in prokaryotic cells, but also inside of the mitochondria and chloroplasts in eukaryotic cells. And the function is for protein synthesis, meaning making proteins. Then if we have a look at a vacuole, so we're thinking about a permanent vacuole that you get in plants here. This structure is filled with fluid and it's surrounded by a single membrane, which we can see here as this darker layer, which is known as a tonoplast. The function is by having liquid in the centre, it pushes everything outwards and that makes the cell turgid and therefore it provides support. It can also be a temporary store of sugars and amino acids, and the pigments that may colour petals to attract pollinators can be found there as well. Next then, chloroplasts, which again are found in plants. This is very similar in structure to a mitochondria. It's also double membrane bound, but this time we have the outer membrane and the inner membrane directly next to each other, both creating a sort of oval shape. Instead, these folded stacks are additional membranes called thylakoids. So we've got these folded thylakoid membranes that fold up to create almost like pound coin or disc-shaped stacks. And those stacks are known as granum for singular or grana for plural. You learn more about that when you get on to topic 5 for photosynthesis. You also have a fluid-filled section here, which is known as the stroma. And the function is photosynthesis. Cell walls, which are found in plants and fungi, but also in bacteria, but we'll come back to bacteria in the prokaryotic cell section. So the cell wall in plants is made of cellulose. The cell wall in fungi, which are actually also eukaryotic cells, is made of chitin. And whatever it's made of, the function is the same. It provides structural strength to the cell. Plasma membranes, this is found in animals and plants. In fact, it's found in all living cells. So it's the phospholipid bilayer, which we can see here. We've got one layer of phospholipids a second. We've got the hydrophilic heads on the outside, hydrophobic tails pointing inwards. And within the plasma membrane, we also have proteins embedded. We have some carbohydrates on the outside as well, and you also have a lipid called cholesterol embedded within. And the function of the plasma membrane is to control what can enter and exit the cell. And this comes up again later in this video when we look at transport across membranes. So that's the eukaryotic cells and their organelles. Next, let's have a look at prokaryotic cells, for example, bacteria. And we can see a range of the organelles here. Some on here you don't need to know. But you do need to know that it has a plasma membrane, a cell wall. Sometimes it has a capsule. You don't need to know the pili for AQA. We've got a flagellum here, which some of them have, and some of them may have one or more plasmid. You do need to know that they have cytoplasm, ribosomes, and then the DNA is as a loop of DNA in the cytoplasm. So let's have a look at that information. The key differences then are between a prokaryotic cell and a eukaryotic cell. Prokaryotic cells are much smaller. In exams, they insist that you say much smaller, not just smaller. They don't have any of the organelles that have membranes, and we describe that as membrane-bound organelles. They do have ribosomes, just like eukaryotic cells, except they're smaller in size, they're 70S. They do not have a nucleus. Instead, their DNA is found as a single circular DNA molecule in the cytoplasm. They do have a cell wall, but it's made up of a glycoprotein, not cellulose, not chitin, and that glycoprotein is called murine. Some of them might also contain any of these three extra structures. They might have one or more plasmids, which are small loops of DNA that only have a few genes on them, often the genes for antibiotic resistance. Some of them have a capsule around the cell wall as well. This prevents the cell from desiccating, meaning drying out, and also it helps to cover the antigens to evade the white blood cells. And they might have one or more flagella, which is for movement or locomotion. Viruses, these are not classed as living, so they're non-living and they are acellular. This has come up as an exam question before to define what those two terms mean. So non-living, it's classed as not being alive because it doesn't do any metabolic reactions within the structure or within that particle. It cannot move, it cannot respire, it cannot replicate without using a host, and it has no means of getting nutrition. It's called acellular, meaning it's not a cell, basically, and it's not classed as a cell because it doesn't have a cell membrane. Viruses are even smaller than bacteria, and they only contain three structures. They contain the genetic material, capsid, and attachment protein. The genetic material is this here in the middle, and that can be DNA and it can be RNA. It'll be one or the other. That genetic material is surrounded by a protein shell called the capsid, not to be confused with the capsule that some bacteria have. So capsid is the protein shell that protects the genetic material. And then the only other structure that all the viruses have are these attachment proteins on the outside, and that's what they use to attach to receptors on host cells. Now we can see additional structures in this picture. We've also got an envelope, and some viruses do gain a lipid envelope by taking it from the host cell that they infect. They aren't able to produce their own one. So that is the three things that all virus particles contain. And that's what we call them, a virus particle, because it's not a cell. So next then is looking at how you can study cells and how this has evolved so that we've been able to understand these organelles that different cells have. And you can use a microscope, cell fractionation and ultracentrifugation. These are things that we're about to go through. The types of microscope, magnification calculation, calibration calculations linked to this idea of the Eyepiece Graticule, and then cell fractionation and ultracentrifugation. Starting with these two key definitions, knowing what's meant by magnification and resolution. Magnification of a microscope refers to how many times larger the image size is compared to the actual size of the object you are viewing. Resolution of a microscope is the minimum distance between two objects in which they can still be viewed as separate, and therefore you get a clear, crisp image still. The resolution of any microscope is determined by the wavelength of the energy source. So for an optical microscope, light, visible light is the energy source, so the wavelength of visible light is what determines the resolution. For an electron microscope, beams of electrons is the energy source, so the wavelength of electrons is what determines the resolution. So if we have a look then at some of these differences between optical, which are the light microscopes, and electron microscopes, which can be scanning or transmission. First then is what it is that creates the image. So for light or optical, it's a beam of light, which is condensed to a fine point using a glass lens. An electron microscope, it's a beam of electrons, which is condensed to a fine point using an electromagnet. Optical microscopes have a much poorer resolution, and that's because the wavelength of light is longer. Electron microscopes have a much higher resolution or resolving power, and that's because electrons have a shorter wavelength. Optical microscopes have a lower magnification, and electron microscopes have a higher magnification. Optical, you can get colour images, whereas you can only get black and white images with an electron microscope. You can then artificially add colour to that image using different Photoshop kind of programs, but with the actual microscope, you only get black and white. And then optical microscopes, you can view living samples, but you can't in an electron microscope. And that's because when you're preparing the sample, it all has to be inside of a vacuum, meaning there is no air. And that's because the air would absorb the electrons and then you wouldn't get an image. So it has to be a vacuum. But because it's in a vacuum with no air, that means you can't create samples with living things because they would die. So just to see an image here, we've got what we can see with an optical microscope. Because they have a much poorer resolution, that means that we can't see the small organelles in a cell. We can see the cell membrane, not in detail, but we can see it's there, cytoplasm and the nucleus and the chromosomes when they condense, but we can't see all of the organelles and we cannot see inside the organelles, but we do get a colour image and we can look at living cells. With an electron microscope, the electrons, as we said, can be absorbed by air, so it has to be prepared in a vacuum, which is why we can't have living samples and it's why it's black and white. A transmission electron microscope, these are the ones where the electrons transmit, meaning pass through the specimen. And that is why we have to have extremely thin slices of the specimen. They are stained and placed in the vacuum, and an electron gun produces a beam of electrons that passes through the specimen. Some parts of the specimen absorb the electrons and appear darker. So that's how we get these different grayscale, black and white parts of the image. So you end up with a 2D image, but you can see inside of the organelles. So this here is a chloroplast, and we can see the membrane, we can see the thylakoids, we can see those stacks, the grana, so you can see details internally. A scanning electron microscope, the specimen does not need to be thin because the electrons are not passing through the specimen. Instead, the electrons are beamed onto the surface and the electrons scatter back. And because they scatter back in different places because of the contours, you end up with these 3D style images. You can't see the internal structures because the electrons bounce back or they scatter back, but you can get an idea of the contours creating a 3D image. The math skill linked to this then is magnification, and it's being able to calculate the magnification, or you might have to rearrange the formula to work out the actual size. So the key thing is knowing the formula. I always remember it as I am, so image equals actual size times magnification, and then I'd rearrange it if I needed to work out magnification. And you usually have to do unit conversions with this because most organelles, the actual size will be measured in micrometers because they're very small structures. But when you're measuring your image size, that is usually going to be in millimeters. So that means when you do your calculation, let's say we're working out the magnification and we're doing image size divided by actual, they both need to be in the same units. So you'd either need to convert your millimeter image size into micrometers, and to convert millimeters into micrometers, you would times by 1,000. Or you'd need to convert your actual size, which would be in micrometers, into millimeters, and that'd be divided by 1,000. Now you could also be asked to calculate the actual size, and in which case we need to swap actual and magnification here. So actual size would equal image size divided by magnification. And if they want you to work out actual size, they'll probably state that they want the answer in micrometers. So again, you might need to do a conversion. Then we move on to what is the eyepiece graticule. So inside of the eyepiece, which is this part here, there is a scale on a glass disc, which is called the eyepiece graticule. So when you look through the eyepiece, you will see what looks like a ruler. And this can be used to measure the size of objects that you are viewing underneath the microscope. But you need to know what each of these divisions on this graticule or ruler is worth. And that is going to vary depending on what magnification you are viewing the specimen at. So that's why we have to calibrate the eyepiece graticule every time we change the magnification. And when we say calibrate, what that means is work out what each of these divisions is worth in terms of a distance. So the way that we would do that is you have your eyepiece graticule in the eyepiece, but you also place a stage micrometer on the stage, which is this bit here. And a stage micrometer is typically 2 millimeters long and it has subdivisions 10 micrometers apart. So it's a glass slide that already has known distances etched onto it. So you line up your stage micrometer on the stage of the microscope, and you would maybe need to twist the eyepiece a little bit so that the eyepiece graticule is now completely lined up next to it. And we then need to look at how many divisions on the eyepiece graticule fit into one division on the stage micrometer. And we know that one division on the stage micrometer is worth 10 micrometers. And we can see that we've got 2 fit into one division. So that then means if that is worth 10 micrometers, we need to do 10 divided by 2, which is 5. And that tells us that each of these small divisions on the eyepiece graticule is worth 5 micrometers at that particular magnification. So then anything you view, you can use your eyepiece graticule to measure the size. And then we know that the number of divisions times by 5 is the actual size. And then if you're going to go up and change the lens to a different magnification, you would do this process again to work out what one of these divisions is worth on the eyepiece graticule. Next is cell fractionation, and this is used to isolate different organelles so that those organelles can then be studied. And this has helped us to identify organelle structures and functions. So there's 2 stages to this. We do homogenization and then ultracentrifugation. And homogenization, this is when the cells are broken open to release the contents, and the organelles are then separated by ultracentrifugation. But throughout this process, the cells must be in a particular prepared solution. It has to be cold, isotonic, and buffered. And you need to be able to explain separately for all of these points why it has to be cold, then why it has to be isotonic, and then why it has to be buffered. So because you're breaking open the cell, that means you're going to have some organelles now exposed to enzymes that they might not usually be exposed to in the cell because we've broken it all open. And those enzymes might damage the organelle before we can examine them. So we make sure it's very cold, it's ice cold, to reduce enzyme activity, to prevent any enzymes potentially damaging the organelles before we can view them. It has to be isotonic, which means the water potential of the solution is the same as the water potential inside of the organelles. And this is to make sure that there is no osmosis, because we don't want any water moving into the organelles because that could cause them to burst. And we don't want water moving out because that could cause them to shrivel. And then it has to be buffered, meaning you use something to maintain the pH, because we don't want the pH to change because that could damage the organelles. And the key thing I've underlined each time is organelles. If you say damage to the cell, that is incorrect because we've already broken open the cell. The cell doesn't exist anymore. It's the organelles that we need to now make sure they're undamaged so that we can go on to examine them. Okay, so a bit more about this homogenization process, basically like a blender. And in fact, when I've done this in lessons, that is literally what we used, a blender. So homogenization is when you break open the cells, and we do that with an ice cold isotonic buffered solution. Then you have to filter your solution to remove large cell debris. And then that filtered solution can be spun in a centrifuge at varying speeds, starting at low speeds. And this is going to separate organelles out from that solution according to their densities. So that's what we can see here. Here is our original solution that we've filtered. The centrifuge will spin, and the centrifugal forces cause pellets to form at the bottom of the most dense organelles. So the first time we spin it, we start at a low speed, and then we increase the speed each time. But our lowest speed, that is going to cause the nuclei to sink to the bottom in the first pellet because the nuclei are the most dense. You then remove the pellet and you then put the rest of that liquid or the supernatant into another tube, spin it at a slightly higher speed. And this time you'd get the chloroplast if you're using plants, and also the mitochondria because they've got similar density. Again, remove the pellets and the supernatant can be spun again. Next then it'd be lysosomes, after that endoplasmic reticulum, after that the ribosomes. And you do need to remember that order. They have done exam questions where they've asked, how would you isolate mitochondria? And for the mark, you needed to know it'd be on the second spin. So chloroplasts and mitochondria are both on the second. We then move on to 3.2.2. All cells arise from other cells, or in other words, we're looking at the cell cycle and mitosis. Now, not all cells are able to continually divide in multicellular organisms, but the ones that can go through the cell cycle. And in eukaryotic cells, when they enter the cell cycle, they will then divide by mitosis or meiosis. Prokaryotic cells replicate by binary fission, and viruses do not undergo cell division because they are acellular, meaning they don't have a cell to divide, and they are non-living. Viruses can still replicate though, but only once they are inside of a host cell, which is the cell that they are infecting. And they have to invade by injecting their nucleic acid into the cell to replicate the virus particle there. And that comes up later in this topic in immunology. So binary fission then, we said this is how prokaryotic cells or bacteria replicate. And this involves, first of all, we have the circular DNA and the plasmids, if they have plasmids, will replicate. And that's what we can see here. We've got circular DNA and we've got plasmids. That is going to replicate. The cytoplasm splits to then create two daughter cells. And each daughter cell has one copy of the circular DNA, but the plasmids, if they are present, don't necessarily split equally, so they might get different numbers of plasmids. It's shown here that they have the same, but it could be different. So then let's have a look at the cell cycle. There are three key stages: interphase, nuclear division, and cytokinesis. Interphase is the longest stage, and we can see that here. Interphase is shown here as G1, S phase, and G2. So we can see about 90% of the cell cycle is interphase. And this is when the organelles double, the whole cell grows, and also DNA replication occurs. So G1 of interphase, that is the growth phase. So that is when we're going to have the growth of the cell and also some of the organelles doubling. S phase is when the DNA replicates. And G2, we have more growth. Also, we have checks for any DNA replication copying errors, and we have final preparations for mitosis, or it could be meiosis. And then this stage is our nuclear division, which can either be mitosis or meiosis. In this topic, topic 2, we only look at mitosis, and that is when you create identical diploid cells. Diploid, di meaning two. Diploid cells are ones that have two copies of the chromosomes. Meiosis, which you learn in topic 4, you end up with 4 genetically different daughter cells are haploid. Haploid meaning one copy of each chromosome. Mitosis creates cells with identical DNA, and therefore it's useful for growth and repair, whereas meiosis creates gametes, which are all genetically different. The final stage, which is just shown as this red line here, it's a very, very short stage, is cytokinesis. And this is when we get the division of the cytoplasm and the cell membrane to create the new cells. So we're going to focus on mitosis, which is just one of these stages in the cell cycle. And mitosis is split up into multiple steps as well. We have prophase, metaphase, anaphase, and telophase, or PMAT, to remember the order. Key facts about mitosis: you only have one division occurring, so you go from having one parent cell to two daughter cells. You get identical, genetically identical cells. They are diploid, meaning two copies of each chromosome. We've only got one chromosome shown here, but we can see we've got the red copy and the blue copy. That's in the parent cell, and it's the same in the two daughter cells. And as we said, the function, growth and repair, and we're going to see that later in this topic when we see clonal expansion in immunity. So prophase, this is our first stage. What you need to know throughout mitosis is what is happening to the chromosomes. In the spec, they describe it along the lines of the behaviour of the chromosomes. So that's what we're going to focus on. What's happening to the chromosomes and why? So in prophase, the chromosomes condense and that makes them visible. So that is what is happening. And in animal cells, these centrioles will also move to be at the opposite poles, which means opposite ends. The centrioles are responsible for creating the spindle fibres, and because they move to both ends, they start to releasing it from both poles, and they'll start to attach to the centromere on chromosomes and the chromatids on the chromosome in later stages as well. Plants have spindle apparatus, but they don't actually have centrioles. Metaphase. So in terms of what's happening to the chromosomes will now line up in the middle of the cell, which is known as the equator. Spindle fibre that is released from the centrioles will now attach to the centromere of these chromosomes, and that's what causes them to go in this single file line at the equator. In anaphase, the behaviour of the chromosomes is now that they'll be split apart at the centromere, and then each chromatid is pulled to the opposite poles of the cells. So those spindle fibres that were produced start to retract, and that is what pulls and splits or divides the centromere in two, so that those individual chromatids are pulled to the opposite poles of the cells, so they're now separated. And once they are separated, we can now call those chromatids, once they're at the opposite poles, chromosomes again. This stage does require energy in the form of ATP, which is provided by respiration in the mitochondria. Telophase is our last stage. The chromosomes are now at each pole of the cell and they're going to uncondense. So they become longer, thinner and no longer visible. Spindle fibres will start to disintegrate, the nucleus starts to reform, and then the very last step, which is not in mitosis but it is in the cell cycle, is cytokinesis, which is when the cytoplasm splits, so does the cell membrane, so you end up with two genetically identical daughter cells. You could be asked to calculate the mitotic index, and this is a way of looking at the proportion of cells that are currently undergoing mitosis compared to the number of cells present when you're looking at a sample underneath a microscope slide, which links to required practical too. So in order to calculate the mitotic index, in your field of view, which means the section that you can see when you look through a microscope, you need to count how many cells are currently in mitosis. The way that you can tell that is whether you can visibly see chromosomes, which is squiggly lines. If it just looks like grey, or in this case it's stained purple, if it just looks like dots almost and you can't see any squiggly lines, that's an interphase, so that is not in mitosis. And most cells, if you remember, we said 90% of the cell cycle is interphase, which should mean about 90% of the cells will be in interphase, and only about 10% will be currently going through mitosis. So it'd count how many cells are in mitosis, divide that by the total number of cells present, and that gives your mitotic index. You might get questions on how can you standardize this. So you could say you're only going to count cells in which you can see the entire cell in the field of view. Now, uncontrolled cell division comes up again later when you learn more details about cancer in topic 8. But mitosis is a highly regulated process, meaning it's very controlled to make sure that cell division is accurate and it's only happening when it's needed. But disruptions to this control do occur, and that then results in uncontrolled cell division, which means you're going to get lots and lots of cells being made, even if you might not need them. And that is how you get a tumour forming, which is just a mass of cells. And those tumours could be benign, which means they're not cancerous, or malignant, which means they are cancerous. And because these tumours are formed because of the uncontrolled cell division of mitosis, many cancer treatments will target mitosis to try and control the rate. Now, this mainly comes up as application questions for topic 4. You learn more theory on this in topic 8 in year 13. 3.2.3 is the next part of topic 2, transport across cell membranes. So this takes us back to the plasma membrane, the cell membrane, which could be a cell surface membrane, but also an organelle membrane. So when we talked about the chloroplast being a double membrane bound organelle, they have one of these membranes. Well, they have two of them because it's double membrane bound. So the membrane is described as a fluid mosaic model, and that's due to the fact that it's a mixture of different molecules. That's why it's a mosaic. So it's made up of the phospholipid bilayer. We've got proteins embedded within it, but also just pinching in externally. We have glycoproteins and glycolipids, and it's described as fluid because there is some slight lateral movement of those phospholipids. All of these molecules are arranged within that phospholipid bilayer, and that is what creates the partially permeable membrane, which is that property that we talked about, or the function. It can select or control what can pass through the cell surface membrane. So let's just go through this in a bit more detail. The main component of the membrane is phospholipids, and this links back to topic 1, which if you haven't already seen my entire topic 1 video where I go through lipids, or if you haven't seen my lipids video in detail, then just search Miss Estruch lipids or Miss Estruch phospholipid bilayer, Miss Estruch entire topic 1, you'll find it. So the phospholipids align as a bilayer, bi meaning two, so 2 layers, that's what it means. And we have the hydrophilic heads on the outside, the hydrophobic tails are on the inside. This is actually zooming in on one phospholipid, and we've got these structures here that need labeling. So 1 is the phosphate group, 2 is the glycerol, 3 and 4 are both fatty acids, but 3 is a saturated fatty acid, 4 is an unsaturated fatty acid because of that double bond between the 2 carbons. A is the hydrophilic head. Hydrophilic because of this negative charge on the phosphate group. And then B is the hydrophobic tail. Those fatty acids are non-polar, so they are hydrophobic, meaning they will repel water, whereas hydrophilic, hydro meaning water, hydrophilic heads will interact with water. So that's why phospholipids in aqueous solution or aqueous environments will create this bilayer. The heads can interact with any of that solution, the water-based solution on the outside, but it will repel the tail, so they spin inwards, and that's how you get this bilayer. Now this goes towards making this partially permeable membrane property because molecules can only move through the phospholipid bilayer by simple diffusion if they can dissolve in lipids and if they're small enough. So it has to be a lipid-soluble substance, which means it has to be non-polar, so it can't be charged. And that's going to be some hormones like estrogen, and also very small molecules like carbon dioxide and oxygen gas are able to. Molecules that can't pass through the membrane, and by that I mean dissolve through the phospholipid bilayer by simple diffusion, is anything that is polar or water soluble, so it has a charge. because that will be repelled by those phospholipids. So things like sodium ions. Or any molecule that is too large to fit through the gaps, so glucose, for example. Now, you do have other components in the cell membrane, making that mosaic feature of the fluid mosaic model. So cholesterol is another type of lipid, and that's present in some membranes in varying amounts. And cholesterol restricts the lateral movement, meaning the sideways movement of other molecules in the membrane. So that's going to be useful as it makes the membrane less fluid at high temperatures, and we want that so that it can prevent water and dissolved ions from leaking out. You also have proteins in your membrane, and proteins can be embedded across the cell surface membrane, or they could be just on the outside. So if it's embedded across, it's known as integral. If it's just on the outside, it's known as peripheral. Sometimes that's intrinsic and extrinsic. So the peripheral proteins, those can provide mechanical support, or they can connect to carbohydrates to make glycoproteins, and carbohydrates can bind to the lipids to make glycolipids. And those are normally, the function is for cell recognition as receptors. The integral proteins are going to be carrier proteins and channel proteins, which are involved in active transport, co-transport, and facilitated diffusion, which is coming up shortly. Protein channels, which we can see one here, these form tubes. We can see it's like a hollow-like tube, and that fills with water. That will enable any of our polar substances, so they're going to be water soluble, to dissolve in that water, and then they can move through the membrane in that way because they can't dissolve in the phospholipid bilayer. Whereas carrier proteins, these will have a molecule bind with them, such as glucose or amino acids, and when that molecule binds to the carrier protein, it causes it to change shape, and that then releases it on the other side of the membrane. Which leads us into the 5 key ways that molecules can transport across these plasma membranes. We have the top 3, which don't require energy: simple diffusion, facilitated diffusion, and osmosis. And the bottom 2 are both active types of transport, meaning they require energy from ATP from respiration, active transport and co-transport. So let's go through all 5 of these. Now, one thing that they'll all have in common is there are certain adaptations to increase the rate at which those types of transport happen. So you could be asked to identify adaptations that cells have for rapid transport. And the sorts of things you need to look for are features that increase the surface area. So for example, microvilli, which is highly folded membranes, increases the surface area. Or if it's a type of transport that goes through a protein channel or a carrier protein, then there might be more of those proteins embedded in the membrane. So again, you've got a larger surface area for that to happen. If it's a cell where there'll be a lot of active transport or co-transport, because those require ATP from respiration, you'll also have many mitochondria as an adaptation as well. So let's have a look at simple diffusion. This is the net movement of molecules from an area of higher concentration to an area of lower concentration. So it's going down the concentration gradient. And that will occur until equilibrium is reached, meaning same concentration in both sides. This process doesn't require any additional energy from ATP. The molecules do still have energy because they're moving, but this is due to kinetic energy that they possess, and that's what enables them to constantly move in fluids, whether that's gas or liquid. For small molecules to diffuse across a membrane, they must be lipid soluble and small. So that is the criteria for simple diffusion across a membrane. It has to be lipid soluble, so that would be a non-polar molecule, meaning it doesn't have a charge, or a small molecule. Facilitated diffusion is still going down the concentration gradient. So it's still a passive process. No extra energy is added in the form of ATP. It's going down the concentration gradient, but it's for any molecule that doesn't fit the criteria we said here, lipid soluble and small. So it's going to be for molecules that are polar, so that means they have a charge, they are not lipid soluble, they're water soluble, and they are too big, or they might be too big. So this is going to happen through either protein channels or protein carriers. It can be through either of those types of membrane proteins for transport. If it's a protein channel, this is the ones where we have the tube-like center filled with water. That enables the ions or polar molecules, water-soluble molecules, to dissolve and then pass through the membrane through that tube filled with water, that protein channel. It is still selective though, as the channel proteins will only open in the presence of certain ions when they bind to the protein. So it is still selective transport across the membrane. Facilitated diffusion can also happen with carrier proteins, and in this case the molecule will bind to the carrier protein, such as it could be glucose, and that causes a change in shape to that carrier protein, and that change in shape causes it to release the molecule to the other side of the membrane. Osmosis, so this is still passive, meaning no extra ATP or energy is needed, and it is going down a concentration gradient. But we need to use the terminology water potential, not concentration gradient, for osmosis to get the marks. So for this, water potential is the pressure created by water molecules, and it's measured in kilopascals and represented by this symbol here. This means water pressure. Pure water has a water potential of 0. So 0 is the highest water potential you can ever get, and that's for pure distilled water. As soon as you dissolve any solutes in it, you get a more negative, or you get a negative water potential. And the more negative a water potential is, that indicates more solutes are dissolved in it. So that's what we mean by water potential. And osmosis is the net movement of water from an area of higher water potential, even though it will still be negative unless it's pure water, but it's a higher water potential to a more negative or a lower water potential, and that's the direction that the water will move, and it's across a partially permeable membrane. So water is the thing that moves, not the solute, and it's always moving towards a more negative water potential, which means more solutes are dissolved in the water at that location. Now, you could get questions linked to this, or mainly actually links to the required practical to do with plant tissue, but the terms isotonic, hypotonic, and hypertonic. Isotonic means the water potential of a solution is the same as the water potential in a cell. And because there's no difference in the water potential, you don't have any osmosis happening, or there's no net movement of water, so the cell will stay exactly the same size and shape. A hypotonic solution is when the water potential of the solution is more positive, meaning closer to 0 than the cell. And that means there's more water in the solution than the cell. So water is going to move into the cell by osmosis. And in animal cells, that can cause it to swell and even burst. Hypertonic solutions is when the water potential of the solution is more negative than the cell, and that will cause water to leave the cell by osmosis, and the cell will then shrivel up. Next, we move on to our active types of transport. So we have active transport first, and this is the movement of a substance from a low concentration to a high concentration. So it's going against the concentration gradient. To do this, it has to use metabolic energy, so that means ATP from respiration, and also it always goes through a carrier protein. So you have to state it's a carrier protein for active transport. So the way that this happens then is, step number 1 is the transport is going through, or transport is occurring with a carrier protein, and those are ones that are going to span the entire cell membrane. A molecule will bind to a receptor on that protein, complementary in shape. ATP will then also bind to the carrier protein from the inside of the cell. So we can see here in the cytoplasm on the inside, it then binds, and it's hydrolyzed into ADP and PI. That releases energy, and that is what causes the carrier protein to change shape and release the molecule to the other side of the membrane. This inorganic phosphate ion was attached the whole time after ATP has been hydrolyzed, and that then gets released, and that causes the protein to return to its original shape. So in order for active transport to continue to occur, you need a constant supply of ATP from respiration. Next then is co-transport, and this is a type of active transport because one of the steps in the process is active transport. So to absorb glucose from the ileum lumen in the gut, which we'll see in topic 3 in digestion and absorption, because there's a higher concentration of glucose in the cell than in the ileum, it's hard to get the glucose to move in because it's going against a concentration gradient. So that's why we need a type of active transport, which is co-transport. So the first thing that happens is actually here. Sodium ions from within a cell are actively transported out of the cell and into the blood in capillaries. That then reduces the concentration of sodium ions in the cell. And as a result, we have now created a concentration gradient from the ileum to the cell. So sodium ions are able to move from the ileum into the cell down a concentration gradient. But they are an ion, they're sodium ions, so they have to do this through a protein. And the protein that they do this through is a co-transporter protein, which means it transports 2 molecules at the same time. And it transports the sodium ions plus glucose, or it could be amino acid as well. That then gets the glucose into the cell. And then we have a high concentration of glucose in the cell compared to in the blood. So glucose can now move down its concentration gradient from the epithelial cell into the blood and the capillary by facilitated diffusion through a protein. And it has to be facilitated because glucose is too large to move by simple diffusion. Next then is 3.2.3, cell recognition and the immune system. So the first part is all about cell recognition and identifying what are self cells, meaning part of your body, and non-self cells, which are not part of your body. And your immune system has cells to identify the presence of pathogens, which are non-self cells, and other non-self cells, which are potentially harmful. And then your immune system will either destroy those or neutralize them to prevent harm. So those cells are the lymphocytes, and each type of cell has a specific molecule on its surface to identify it. And those molecules are usually proteins, and because they're proteins, they have a unique 3D shape. There's overlap here with biological molecules from topic 1 because common marking points linked to this are stating that it has a tertiary structure because it's a protein and that's what gives a unique 3D shape, but tertiary structure would be the key marking point, and that would be complementary in shape to, and that's actually going to link to some of the cells that we learn about later, but it's the idea of tertiary structure, complementary in shape. That's how you can work out if it's a self or non-self cell, and a bit more on that when we get to the T and B cells. But thinking now more about what counts as a non-self cell. So if a non-self cell is detected, a response will be triggered to destroy that cell. And these are the 4 examples that you need to know that could trigger an immune response because they're detected or could be detected as non-self cells. So number 1 is a pathogen. So bacteria, fungi, they'll have antigens on their surface that can be used to identify them as non-self cells. It could be cells from another organism of the same species. So for example, if you've had an organ transplant, that could be detected as non-self. And we don't want that to happen because then your immune system will start to attack and destroy the cells from that transplanted organ. It could be abnormal body cells. So for example, cancer cells, their antigens on the surface will slightly change. They now don't get detected as self cells, they're now detected as non-self. Or it could be toxins because some pathogens release toxins into the blood, such as cholera. So I use this word antigen quite a bit, but an antigen is a foreign protein that generates an immune response by the lymphocytes when it's detected, and it's located on the surface of cells. That is quite a common 2-mark question. You need both of those points to define what an antigen is. Antigens can change though over time, and this links to antigen variability. So a pathogen's DNA can mutate frequently, and if the mutation occurs in the gene that codes for the shape of the antigen, then that shape of the antigen will change. And therefore any previous immunity that you might have had for that pathogen, based on the recognition of the old shaped antigen, it will no longer be effective because the memory cells have a memory of the old antigen shape. So this is known as antigen variability. And for example, the influenza virus, which is the virus that results in the flu, that mutates and changes its antigens very quickly. And that's why you need a new flu vaccine every single year. So let's go on to the immune response then. If you do have this non-self-cell detected, we said an immune response occurs. So if a pathogen gets past any chemical or physical barriers, which you don't learn for AQA for A-level, but that's things like your skin barrier, your stomach acid, and it can then enter the blood, then this is when the white blood cells are the second line of defense, and you either get a specific response by the lymphocytes or a non-specific response. And in fact, you get both, starting with the non-specific response of the phagocytes and then the lymphocytes. So the non-specific response by the phagocytes is phagocytosis. And a phagocyte is a type of white blood cell. For example, a macrophage is an example of a phagocyte. And phagocytes are white blood cells that can do phagocytosis. They're found in the blood, but also in the tissue. And phagocytosis is a non-specific immune response. What that means is any non-self cell that is detected, and that means any of those 4 items that we listed a couple of slides ago, if they are detected, it will trigger phagocytosis to happen every single time. So phagocytosis then, the first thing that happens is our phagocyte, for example a macrophage, which is in the blood or in the tissues, and any chemicals or debris that are released by the pathogen will attract the phagocyte to move towards the pathogen. So that's how the phagocyte is able to detect where the pathogen is. Now on the phagocyte, there are many receptors which are binding points on the surface of that phagocyte that can then bind to the antigens or any chemicals on the pathogen. The phagocyte, once it's then bound to it, the phagocyte changes shape, which is known as engulfing. So it completely surrounds and engulfs the pathogen, and it then will put it inside of a vesicle inside of the cell. And that vesicle is known as a phagosome. So we can see here the pathogen's been engulfed and it's now trapped inside of the phagosome. You then have the lysosomes within the phagocyte will move towards, so the lysosome shown here in blue, it will move towards and fuse with the phagosome, and it will then release its contents. And the contents of a lysosome is digestive enzymes. And in this case, they're known as lysozyme enzymes. They're digestive, which means they're lytic, or they're going to split, they're going to break apart, hydrolyze the pathogen. So that is how the pathogen is then destroyed. From the broken down, hydrolyzed, digested pathogen, any soluble products that are going to be useful can be absorbed by the phagocyte, and then the antigen is placed on the surface of the pathogen, and that is going to be used later on as a way to signal to other white blood cells known as lymphocytes that a pathogen has been detected and destroyed. So that takes us on to the first type of lymphocyte we're going to go through, T lymphocytes, or T cells for short. These lymphocytes are involved in the specific immune response, meaning they respond in a particular way based on the particular shaped antigens that are detected. Lymphocytes are made in the bone marrow, and this is extra information, you don't need to know it, but they're called T lymphocytes because T cells then go to mature in the thymus, which is this structure here in the body. And they are involved in the cell-mediated response. So this is where we look at how the antigen-presenting cells are going to help trigger the cell-mediated response. And the antigen-presenting cells could be, like we just said, a phagocyte, for example, a macrophage, which has engulfed and destroyed a pathogen and then it presents it on its surface. But it's actually any cell that has a non-self antigen on its surface. So that could be one of your own body cells that's become infected. It could be one of those phagocytes presenting the antigen on the surface. It could be cells of a transplanted organ. It could be cancer cells. And because they have antigens presented on their surface, that is going to enable lymphocytes to respond to those antigens in this cell-mediated response. So let's carry on from this bit we were at. So once the pathogen has been engulfed and destroyed by the phagocyte, antigens are then presented on the cell surface. It's now called an antigen-presenting cell. And we can see an example of that here. We've got an antigen-presenting cell. One type of T lymphocyte is known as a helper T cell. And helper T cells, they have these receptors on their surface, and that can attach to complementary shaped antigens on antigen presenting cells. Once it then attaches, that activates the helper T cells to divide by mitosis, and that will then replicate and make large numbers of clones of that exact helper T cell that had the receptor complementary in shape to that exact antigen. That stage is known as clonal expansion. Clonal because you're making identical copies, expansion because you're making large numbers. And this is where there's that link back to mitosis. We said the role of mitosis is growth and repair. In this case, it's growth and making large numbers of identical T helper cells. Now we've got that large number of identical T helper cells, they actually differentiate into different types of T lymphocytes. So some will actually just remain as helper T cells because those are then going to go on to activate the B lymphocytes, which we're going to go on to shortly. Some of them are able to stimulate macrophages so that they will perform more phagocytosis and engulf and destroy more of those non-self cells. And then some of them become cytotoxic T cells, sometimes called killer T cells. AQA use the term cytotoxic T cells. And a cytotoxic T cell is able to destroy your own body cells that are infected with a pathogen, typically a virus. And the way that they do this is cytotoxic T cells release a protein called perforin, and that protein can embed in the cell surface membrane to make a hole or a pore. And that means that either lots of water will move in and cause the cell to burst, destroying the infected body cell, or it will cause lots of water to move out, causing the cell to shrivel, and again, it will cause cell death. So this is most common in viral infections because viruses can only replicate when they're inside of the body cell. So if you destroy the body cell, the virus can't replicate, and that gives more time then for the phagocytes to locate and destroy the virus before it can replicate and cause more damage. And this is why you get a sore throat when you have a cold. The cytotoxic T cells are destroying the infected body cells in your throat. You're sacrificing body cells to prevent the virus replicating and infecting even more cells and tissues. Now, B lymphocytes, this is the humoral response. Again, all lymphocytes are made in the bone marrow. You don't need to know this, but the B is because it matures in the bone marrow as well. This is a humoral response, meaning within liquids or within body fluids, because in this instance, the B cells will only respond to antigens that are floating within the blood or within body fluids. They don't respond to the antigen presenting cells in the same way as a cell-mediated response with the T cells. So let's have a look at the B cell activation. So you have approximately 10 million different B cells which have antibodies on their surface, complementary in shape to 10 million different antigens. And you have these circulating in your blood, and if you get an antigen in your blood, collide with the complementary shaped antibody on a B cell, that is going to cause the B cell to take in that antigen by endocytosis, which means basically surround and engulf it. So this is responding to the antigens in the blood, not the entire cell. When the B cell then collides with the helper T cells that we talked about in the previous slide, that activates the B cell to go through clonal expansion and differentiation. Now, this can happen because when that B cell takes in the antigen, it's going to break it down, but it can then actually also present it on its surface. So that means it's able to bind to helper T cells that have that receptor complementary in shape to the antigen. That is then going to trigger the B cell to be activated to do the same thing as the T cells. It undergoes mitosis. You get a large number of clones of that B cell that has that exact shape, antigen and antibody on its surface, complementary to that foreign protein, the antigen. But this time, the cells differentiate into different molecules. They can either become plasma cells or memory B cells. The plasma cells, which you can see here, these are the ones that make antibodies. Memory B cells are the ones that remain in your blood for long periods of time, and they can divide rapidly into the plasma cells to release large quantities of antibodies so quickly if you get reinfected with the same pathogen. So they're responsible for you being immune, basically, to particular pathogens and disease. So let's have a look at this. Memory B cells, as I said, they can live in your blood for a long time. They can be decades in your blood, whereas plasma cells are very short-lived. And those are the ones that make the antibodies, so that's why we need to store a memory of that antibody. So if you are reinfected, you can then have the memory B cell rapidly differentiate into plasma cells, which can then make lots of antibody so quickly that it should help to destroy the pathogen before you get any symptoms, which means you are immune. Or it might just mean you get very, very minimal symptoms. And this is known as active immunity. You are immune to that pathogen, and it's active because you have memory B cells against that pathogen. So this links this concept of primary and secondary response. Primary immune response is the first time you are exposed to a pathogen. And because it's the first time you're exposed, it can take a longer period of time for the B cell to collide with that antigen, causing it to engulf the antigen, and then you get the clonal expansion, differentiation. All of that process of the humoral response has to happen. So it takes a longer time to make antibodies, and you don't get as high a concentration. The second time you're exposed to that same antigen, because you now have the memory B cells present in your blood, those can then collide with the pathogen's antigen again and differentiate rapidly into plasma cells. So you end up with large quantities of antibody much, much quicker. And this is why the secondary response should mean that you get no symptoms or very few, because you get the antibodies being produced so rapidly and in large concentrations. So let's have a look at what antibody is. They are proteins that have a quaternary structure, meaning more than one polypeptide chain, and they happen to have 4 polypeptide chains. The end bits here are the variable regions, meaning those are the parts that are going to be different in shape and combine to the different shaped antigens. The red part is constant, meaning it's the same in all antibodies. The heavy chain is this longer one, and you have 2 heavy chains. The shorter chain is known as the light chain. Here are the antigen binding sites. You can bind one here and here. And then you also have these bonds here and the hinged section, which makes the antibody flexible, so it can easily bend and attach to one antigen over here, bend and attach to another over there, which is how we get agglutination happening. And agglutination is the idea that multiple antibodies can bind together lots of different antigens at the same time, so they all clump together. That makes it easier for the phagocytes to, first of all, locate where all the pathogens are, and then they can more efficiently engulf and destroy them. So antibodies, they create these antigen-antibody complexes, and we get agglutination happening. But they don't directly kill the pathogen. They just make it far more efficient for the phagocytes to then destroy the pathogen. So different types of immunity. First of all, passive immunity. Passive immunity is when the antibodies are introduced into the body and the pathogen doesn't enter itself. So you don't actually get any antigen or pathogen in you. So that means the plasma cells and memory cells are not made. You just get a direct source of antibody. So you don't get any long-term immunity because there's no memory B cells. A natural example of this is in breast milk. So through breastfeeding, breast milk contains antibodies. That means babies will have passive immunity whilst they are breastfeeding. And when I say whilst, it's not literally just while they're breastfeeding. The antibodies can last for a short period of time. Active immunity is when you have been exposed to the pathogen or the antigen, and therefore you do have memory B cells in the body. And that could be natural active immunity following infection, or it could be artificial active immunity following a vaccination where you might have a weakened version of the pathogen or the antigens injected in. So vaccines, that means a small amount of either weakened or dead pathogen or the antigens itself introduced to the body. And that could be as a liquid through the nose or mouth, or it could be an injection. And exposure to those antigens activates the B cells to go through clonal expansion and differentiation, just as we saw in the humoral response. So those B cells will then undergo mitosis, make large numbers of identical cells, which will then differentiate into plasma cells and memory B cells. Plasma cells will make antibodies, which will then destroy any of that weakened or dead form of the pathogen. And the B memory cells will then remain in your body for decades. So if you are reinfected with the same pathogen, you can make large numbers of the antibodies so rapidly that you shouldn't get symptoms. Herd immunity is if enough of the population are vaccinated against a particular pathogen. That means that the pathogen cannot spread or transmit amongst the population. And that's because if you have most of the population immunized, that means that they can't transmit the pathogen onto someone else, and therefore hardly anyone in the population is going to have the pathogen. And this provides protection for those who are not vaccinated. So maybe they are too ill to be able to be vaccinated, or maybe they're pregnant, or they have some lowered immunity that means they aren't able to. The next bit is then looking at HIV, an example of a virus, and it is a retrovirus. It has a lipid envelope around it, but that is taken from the host cell membrane. The envelope has the attachment proteins, which are glycoproteins, which is how it attaches to the host cell. So here are the key structures that you have in the HIV particle. You have the core, and that is the genetic material, which is RNA, and it has the enzyme reverse transcriptase as well, which is needed for the virus to replicate. You have the capsid, which is the outer protein coat protecting the genetic material. There is the envelope, that is an extra outer layer, but that is made out of lipids taken from the host's cell membrane. And then you've got the protein attachments on the exterior of the envelope to enable the virus to attach to the host's helper T cell. So let's have a look at how HIV replicates in helper T cells, because those are the host cells that it infects. HIV is transported around in the blood until it attaches to a CD4 protein on a helper T cell. So here is a helper T cell. Here is a CD4 protein, which is a receptor. And we can see that the HIV with the attachment proteins binds to that receptor. The HIV protein then fuses with the helper T cell membrane, and that enables the RNA and the reverse transcriptase enzyme to enter with the capsid, and then it does get released from the capsid. The HIV enzyme reverse transcriptase copies the viral RNA into a DNA copy and moves into the helper T cell nucleus. And this is why it's called a retrovirus, because it's doing the reverse of transcription. You've got an RNA copy being turned back into DNA. The DNA then moves into the nucleus. It combines with the host's DNA, and that is where the mRNA is then transcribed along with the host cell's mRNA to create the viral proteins. The viral proteins are then reassembled, and they'll then move towards the cell surface membrane, and they are released from the cell. And that then goes on to make a new virus particle. Now, the difference between HIV and AIDS is HIV is the virus, and it specifically targets and it destroys helper T cells, because as it replicates inside of them, it destroys the helper T cells, which activates B cells. So if we don't have those helper T cells, they're not there to activate B cells. That means we have now significantly reduced the immune system. And if enough helper T cells are destroyed, then you end up with AIDS, acquired immunodeficiency syndrome, where you've become so vulnerable to opportunistic infections and cancers and you can't defend yourself. So symptoms of AIDS are chronic infections, weight loss, fever, night sweats, and they can be followed by more serious illnesses like pneumonia, tuberculosis, and certain cancers. A person is HIV positive if they're infected with HIV. AIDS is when the replicating virus in the helper T cells interfere with the normal function of the immune system, and it's normally classed by how many of the helper T cells have been destroyed. The last bit of this topic is monoclonal antibodies. Mono meaning one, clonal meaning identical. And a monoclonal antibody is a single type of antibody that can be isolated and cloned. And antibodies are proteins which have binding sites complementary in shape to certain antigens. Now, this is what is created in the immune response, monoclonal antibodies, lots of identical copies of the same antibody. But this has been manipulated to create monoclonal antibodies for medical treatments, medical diagnosis, and even pregnancy tests and other chemical tests. Now, targeted medication, this can use direct monoclonal antibody therapy, and this is when you have some cancers that can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of cancer cells. Those antibodies are then given to the cancer patients and they'll attach to the cancer cells. While the antibodies are bound to the cancer cell antigens, this prevents chemicals from binding to the cancer cells, which enable the uncontrolled cell division. And therefore, the monoclonal antibody prevents the cancer cells growing anymore. And as they're designed to only attach to the cancer cells, they should only affect the cancer cells and not healthy body cells. Indirect monoclonal antibody therapy can also be used, and this is when you would attach a drug to the monoclonal antibody. So in this instance, you've still created the antibody to be complementary in shape to the antigens on the cancer cells, but the cancer drugs are then delivered directly to the cancer cell, and it kills the cancer cell rather than lots of healthy body cells. So this should reduce the harmful side effects that affect traditional chemotherapy and radiotherapy can produce. And this is known as a bullet drug. It's going straight to where it's needed. So if you have a look at medical diagnosis, monoclonal antibodies can be used to test for things like pregnancy, influenza virus, hepatitis, chlamydia, prostate cancer, but also it's not on the spec because this happened after the spec was created, but COVID-19, all of those lateral flow tests that we used to have to do all the time use monoclonal antibodies. And this works via an ELISA test. So an ELISA test, the enzyme linked immunoabsorbent assay, it uses two antibodies. We can see here in the pregnancy test, we've got, first of all, a mobile antibody, and then we have a immobilized antibody. And we actually have a third one here. We've got an immobilized one as well. And in this instance, what happens is you would dip your pregnancy test into the urine that you are testing to see if someone is pregnant or not. Because when you are pregnant, you create this hormone HCG and it can be secreted in your urine. So this is absorbent paper. That means the urine with any hormones dissolved within it are going to move up through that absorbent paper. The hormone is complementary to the mobile antibody, which also has a coloured dye attached to it. So that will then move up the pregnancy test. And at point C, you have immobilised, meaning it can't move, antibody that is also complementary in shape to the hormone. So that will then cause the first antibody that has the hormone attached to it to attach to the immobilised one. And it also has that coloured dye on it. And that's why at that exact position you get a coloured line. Any antibody that didn't bind to the hormone will move past section C, but we do have antibodies also immobilized at point D that are complementary in shape to the end of that antigen. So those will bind and you get a second line. And in this way, whether you have that hormone or not, you should always get the line at D. And that is to help prevent false negatives, because it shows us that you don't have a coloured line here if you don't, but the antibodies are definitely able to move, so the test is working. Whereas if you have a line here, that means you are pregnant because you do have that hormone present. And this line here confirms that the test was working, so antibodies did move through the test. Now you can also have different versions of this. So we've got one here where we would add the test sample from a patient to the base of a beaker or the base of a well, and would then wash to remove any unbound test sample. Then you'd add an antibody complementary in shape to the antigen you are testing for, and that will bind if it's present. Again, wash to remove any that didn't bind. We then add a second antibody that is complementary in shape to the first one, so that's only going to bind if the antigen was present and therefore the first one bound. Again, wash to remove any that's unbound. Then you'd fill the wells with a liquid which is a substrate for this enzyme. And when those react together, you get a colour change. So the presence of the colour indicates that you do have that antigen present, and the intensity of the colour indicates the quantity present. Now, there are some ethical issues around monoclonal antibodies, because creating monoclonal antibodies requires animals such as mice to produce those antibodies in tumour cells, which leads to ethical debates as to whether this use of animals is justified to enable better treatment of cancers in humans and detect disease. Because in order to do this, there will be suffering to the animal, and then at the end they would be killed because they wouldn't be needed. So that takes us to the end of topic 2. And if you did find this helpful, then don't forget you can check out my A-level notes and flashcards for mark scheme specific points. But that is it for today's video. Hope you found it helpful, and I'll see you in the next one very soon.